For many of us, our memories are a source of great joy. They allow us to relive past experiences, both big and small, that have helped make us who we are today. Memories play a key role in our sense of identity, and so it's natural to want to hold on to them for as long as possible. But some memories are painful and can cause a lot of distress, which is why researchers have been working hard to find ways to delete or even create memories. And the results may surprise you. So, what exactly is a memory? To answer this question, let's break them down into two categories. First, there are episodic memories, which are memories of specific events that we've personally experienced. These are the kinds of memories that allow us to reminisce about our past. Then there are semantic memories, which are memories of facts and general knowledge. For example, if I asked you what the capital of France is, your answer would likely be informed by your semantic memories. Episodic and semantic memories are stored in different parts of the brain. Episodic memories are stored in the cerebral cortex, while semantic memories are stored in the hippocampus. But they're both types of explicit memories, meaning that they can be consciously recalled and in many cases verbally expressed. However, not all memories fit this description. Implicit memories, also known as non-declarative memories, are memories that are not stored in the cerebral cortex or hippocampus and cannot be consciously recalled. Procedural memories are a type of implicit memory that involves knowing how to perform a task. For example, if you ride a bike, you have the procedural memory for riding a bike. Even if you couldn't remember ever learning how to ride a bike, you would still know how to do it. This is because the relevant information, such as the sequence of movements involved in riding a bike, is stored in the cerebellum and other parts of the brainstem. Like procedural memories, priming and conditioning are also types of implicit memory. Conditioning refers to learning by association. For example, if you were to ring a bell before feeding a dog, the dog might eventually learn to associate the sound of the bell with mealtime and start salivating when it hears the bell ring. Even if the dog has no conscious memory of being conditioned, this type of learning has had an impact on its behavior. Priming, on the other hand, is a type of implicit memory that involves making mental associations. For example, if you were to read the word doctor, you might be more likely to believe that the word nurse is true. This is because the word doctor activates the concept of nurse in your mind, making it easier to recognize. Again, this happens outside of your conscious awareness. So, what does all this mean for our ability to delete or create memories? Well, to answer that question, let's first talk about how memories are formed. The process of forming a memory begins with encoding, which is the initial stage of acquiring and processing information. During encoding, our brain takes in sensory information and transforms it into a form that can be stored. Next comes storage, which is the retention of encoded information over time. Finally, there's retrieval, which is the process of accessing stored information when we need it. Each of these stages presents an opportunity for researchers to intervene and manipulate memories. In fact, scientists have already made some progress in this area. One way to interfere with the formation of a memory is to block the initial encoding process. For example, if someone suffers from a concussion, their ability to form new memories may be impaired. Similarly, certain medications, such as those used to treat Alzheimer's disease, can interfere with the encoding of new memories. However, blocking the formation of new memories is not the only way to manipulate them. Scientists have also found ways to target the storage and retrieval processes. For example, in one study, researchers found a way to reactivate dormant herpes simplex virus 1, or HSV1, which causes cold sores. The reactivation of this virus caused the release of a protein called ICP0, which binds to the protein CTIP2 and disrupts the storage of new memories. This disruption resulted in mice forgetting odors that they had previously learned to associate with food. While this research was conducted on mice, it suggests that similar mechanisms may be at play in humans. If so, it might be possible to develop drugs that target HSV1 in order to treat memory disorders. Another way that scientists have interfered with the storage of memories is by injecting a drug called anisomycin into the brains of rats. Anisomycin prevents the formation of new proteins, which are necessary for the storage of memories. As a result, rats that were injected with anisomycin were able to forget the location of a hidden platform in a water tank. Again, this research was conducted on rats, but it suggests that similar techniques may one day be used in humans. But what if instead of trying to delete a memory, we could try to create one? This might sound far-fetched, but it's actually already been done. In one study, researchers taught mice to fear the smell of a rose. They did this by pairing the smell of rose with a mild electric shock. Once the mice had learned to fear the smell of rose, the researchers injected them with a drug called BDNF, which promotes the growth of new neurons. The drug was effective at erasing the mouse's fear of the rose scent, but it also caused the mice to form a new memory, namely that the rose scent is not dangerous. While this research was conducted on mice, it suggests that similar techniques may one day be used in humans to treat post-traumatic stress disorder, or PTSD. In another study, researchers injected rhesus macaque monkeys with a virus that contained a gene for a protein called ARC. ARC is involved in the formation of new memories. By injecting the virus into the monkeys' brains, the researchers were able to increase the amount of ARC protein in the monkey's hippocampus. The monkeys then underwent training to remember the location of a cue associated with a reward. The monkeys that had received the injection and thus had higher levels of ARC protein were better able to remember the location of the cue. Although this research was also conducted on monkeys, it has important implications for humans. In fact, scientists have already begun using gene therapy to treat memory disorders in humans. For example, in one clinical trial, researchers injected a virus that contained a gene for the enzyme CHTF77 into the brains of patients with Alzheimer's disease. The patients had a genetic mutation that caused them to produce less CHTF77 than normal. By replacing the faulty gene with a healthy one, the researchers were able to improve the patient's memory and cognition. These are just a few examples of the amazing progress that's being made in the field of memory research. Who knows what the future will hold? Maybe one day we'll be able to delete painful memories or create new positive ones at will. Or perhaps we'll develop effective treatments for memory disorders like Alzheimer's disease and dementia. One thing is for sure, though, the research being conducted today will have a profound impact on our understanding of memory and the human brain. What do you think? Would you ever want to delete a memory? What about creating a new one? Let us know your thoughts in the comments below. And don't forget to like and subscribe for more amazing discoveries from the world of science. Thanks for watching.